do that this morning. No, here we go again. Okay, there we go. Okay, so yeah, so we can make these awesome mutations in here, but one of the things that can happen is we can see methylation patterns that lead to mutation. So you might recall from last week we talked about methylation patterns distinguish the parent strand from the daughter strand, but those are tightly regulated methylations. Okay, occasionally you can see methylation that occurs that is, not where it should be. Okay, so what we end up seeing is that our guanine here can become methylated, give us methylguanine, and then that methylguanine will actually end up base pairing with thymine. So instead of making our GC base pair, it ends up making a GT base pair. And I guess that methyl group is effectively blocking, the cytosine from being in the right space, but thymine is the right size, and it can base pair with the two amine groups. And of course, there's nothing to base pair over here with the methyl group because they're non-aryl, non-labile protons. So what we end up doing is we end up incorporating a mutation in there, so we get an incorrectly paired DNA. Now, of course, our opposite direction, for our lagging strand. So if this is our leading strand in pink, our lagging strand in blue, our lagging strand will be correctly made because we still have our C there, and G will pair with that. It's just here our methylguanine pairs this way, so we end up with a slightly different stuff in there. But that's one way that we can end up with mutations in there. We can also see bases that become alkylated as well too. And then we can directly repair them by actually removing or abstracting that methyl group here. So you can see in this case formaldehyde plays a role. So our methyladenine, that formaldehyde, once we activate the methyl group, typically by adding a hydroxyl group in there, either through an oxygenation reaction or, that's essentially what we got going. Yeah, oxygenation reactions in here. So we oxygenate, oxidize that methyl group, and then that makes it a good leaving group from the amino group, and our formaldehyde can pull it off. And then we're left with our cytosine or adenine to repair it. All right. So what happens if we have a lot of damage? As long as we have a template, a parent template, we can go back and so we can go back to the plans and figure out what it was supposed to be and repair it that way. If we have massive damage or we don't have any DNA that's in there, we can look at using a different chromosome as a template. And this is where we get into our recombination. This is why we can actually have those transposons that can move stuff from area to area. So we can use another chromosome that has a similar gene in there. And then we can also have something called error-prone translesion synthesis, where it's just kind of our best guess. And the analogy I always like there is if like the barbarians come and knock down your door, and you're repelled, but there's another way of coming. And you're going to put up barrels and benches and nail whatever you can up there to fix that. We're going to try and repair that, even though it might not function exactly right. It might be sufficient to allow survival, and that's what we're looking at. Okay, so if we have a lesion in our DNA, we can stall out our replication. So our replication fork gets past here, and then it just kind of stops. So what we need is a recombinatorial DNA repair, where we find similar DNA in a different chromosome and import that and use that as a template. Or we can do this recombinatorial, error-prone repair as well, and just kind of make our best guess there. All right. The same thing can happen with the double strand break as well too. If we have this break here, we just kind of stop because there's nothing to copy. So in that case, we have to use recombinatorial DNA repair. All right. So our error-prone repair mechanism in here in bacteria is part of the SOS response. So if you have extensive DNA damage in here, you will see this error-prone, these error-prone repair mechanism proteins upregulated in expression, and you have all these like UVRA, UVRB, all this good stuff in there too. I want to say like UMU is unmutable, but it always makes me think it was the UMU eyelids thing or something, whatever. That's one of the almost meme-ish emojis now. But anyway, they'll interact with REC-A to give us this DNA polymerase 5, and this polymerase 5 can process past the damage area, and you just make kind of your best guess as to what should go in there. So why do we have this error-prone repair? Well, we're gonna, some cells are just gonna flat out die. Some of them are gonna have massive mutations, but some of them will survive, and that's our goal, survival in there. Okay. We see these in mammals as well too, so it's not just a bacterial repair mechanism. So we have this polymerase eta that will assist whenever we have one of these dimers that halt a replication fork. So if you have like a particular ionizing radiation or UV damage, that you get one of those dimers in there, this polymerase eta will work to go past it. Most of these polymerases will only replicate short regions of DNA, unlike polymerase 3 or polymerase gamma. I think that's the one for mammals that will do thousands and thousands of base pairs at a time. Here we're looking at maybe a few dozen. Okay. All right. All of this leads us into the idea of recombination. And I'm not going to go into excruciating detail about this because there's just way too much. And, it's kind of like swimming in the ocean. You can swim on the surface, maybe do a little surfing or something like that. And you can dive down, see little fishes in there, and you can go down further and further and further until you're in the Marianas Trench or something like that. So we're going to try and keep it near the surface and not go into too much depth here, but just kind of discuss a little bit. So recombination, we can basically take segments of DNA and move it from one section to another section. Usually this occurs within a chromosome, but you can also take it from another chromosome as well. A lot of this can be done for repair, as we've just seen. We can also look at this in terms of segregating chromosomes during meiosis. And it also helps to enhance genetic diversity, so we can get some differences in here. It's like, hey, is it better like this or better like that? If you wear glasses and go see the eye doctor or something. So for any organism that reproduces by sexual means, recombination and mutations tend to be our driving forces of evolution. You may see a particular mutation that arises that confers an evolutionary advantage, and so future descendants will keep that particular mutation. There may be a mutation that inhibits synthesis, and those descendants will most likely die out. And then there are some that are neutral, and it's just what's the difference between brown hair, blonde hair, red hair, whatever. There doesn't seem to be any significant difference for that, so there's no particular reason for it to. I don't know. Getting too far away from where I went. Okay. Well, we have three classes of recombination that we can have. We can have homologous recombination. This is also known as general recombination, where basically we have two DNA segments that share a similar sequence, and that long chain of similar sequence basically means you can just take this one and move it over to where the other one is. Okay. You can have site-specific recombination, where the exchange only happens at a particular sequence. So much like our restriction endonucleases that recognize a specific sequence and make a cut, here we have recombinases that will recognize a specific sequence and transpose that. And then we have DNA transposition, our jumping genes. These are short DNAs that can just move from one chromosome to another. Okay. If we look at homologous recombination, that can help with DNA repair. It can link sister chromosomes to properly segregate them between the parent cell and the daughter cell during meiosis. And of course, we can look at DNA exchange and genetic diversity in there. That's a terrible, terrible explanation, but anyway. For bacteria, and I really kind of want you to know what it is that's going on. But the big key here is, you have this recombinase enzyme that facilitates, catalyzes the reaction. And you actually take these two different regions, and you make this cross structure in here, which is known as a holiday junction, because that's where they party. Okay, no, never mind. It's just named after the person who discovered it. And you'll need a nuclease and a ligase. Okay, so what we end up having, here's our double strand break in here. We see this little break. So we bring in another strand, what we call the strand invasion here. And we end up with this nice little holiday intermediate, where you can see we have this kind of crossover, okay? And then we can cross that back over to give us back to our regular chromosome in there, whatever. Anyway, The REC enzymes are the ones that are involved in recombination in here. So you've got REC A, REC B, REC C, REC D, and they all move together as a big, nice little superstructure in here. We're not going to go into detail about this. I kind of want you to know what they do, but we're not going to talk about them in great detail. Casein. And you can see, much like our replication enzymes, they have different regions in there that play different roles, like unwinding. You got a helicase in there somewhere, nuclease, and our chi sections keep them unwound so that that our rec A can pop on there and do its thing. Okay. Anyway, this is kind of neat that you can actually see rec A bound to our DNA filament in here. All right, I don't really want to talk about those though, right? So again, here we have our two strands, two different DNA strands in here. We get this nice little Hall-Day junction, so you can actually cleave across the junction here, so that you end up having part of the red one now paired with the blue one, and so on and so forth. So you get that recombination in there. For eukaryotes, things are a little bit different. This occurs quite often during meiosis. We want to make our haploid cells in here, so you're going to get our 4 homologous chromosomes. You get a nice little tetrad. So you want 2 pairs of sister ones that are connected at the centromeres. Cells, divide twice, and we separate for meiosis 1 and meiosis 2, and we end up making our awesome little cells here. So that first meiotic division, there you got your pairs there, and then we split off during the second meiotic division to give you the haploid gametes. All right. See my baby. No, never mind. I'm sorry. I'm in a mood. Yeah. Okay. All right. So our recombination links these sister chromatids together, but we're not going to sweat this. Like I say, I just want to fly kind of through this here, crossing over. You guys are probably too young to remember some cheesy live show where some guy claimed he could talk to the spirits of the dead. It's like Crossing Over with John Edwards. So I always think of that here. Anyway, never mind. Okay. All right. So if we can have our DNA recombine, if we can experience this recombination, what does that mean for gene mapping? All right. So we can actually figure out, kind of go do an evolutionary tree and figure out where genes used to be on chromatids and look at a comparison between species as well too, to give us an idea of how genes have moved. All right. So if we look at our various meioses, then we can look at 8 possible haploid gametes or spores, depending on what organism we're looking at here, right? Just on the way that they can recombine. All right. If we end up making a mistake, which is rare, but it does happen, you can end up with something like aneuploidy, where you have the wrong number of chromosomes. You have an extra chromosome, or you're missing a chromosome. We see this with Down syndrome, also known as trisomy 21, because you've got 2 copies of a particular of chromosome number 21, which means there's 3 in there. If you're missing a copy, you have monosomy. So you can also have this, darn it, this Turner syndrome in here. Let's see if I can put that down there. That should work. Okay. Got things overlaying my screen. But basically, if you're lacking a particular X chromosome in there, you get Turner syndrome. But if we just have the wrong number of copies in there, usually an organism may survive, but it's going to have some developmental issues in most cases. Sometimes it can be relatively mild. Sometimes it can be extremely severe and can lead to death or miscarriage early on as well too. Okay. So how do we get aneuploidy? If we look at the, for males, the germline cells undergo rapid meiosis at puberty. So constantly, guys are constantly making sperm, whereas I'm sure you've heard women are, once you hit puberty, you have as many eggs as you're ever going to have in your life. So they have that suspended animation phase in there. So those germline cells undergo meiosis in the fetal stage, and the process stops for somewhere between 13 and 50 years until the egg cells mature. All right. So disruptions to crossovers during this stage can cause aneuploidy, and I always laugh because my wife turned 35 when she was pregnant with our second child. So you hit 35 and it's like, boom, you're a high risk pregnancy. Even though all the other factors were good, just that age, the likelihood of damage increases with age. It's amazing too. There's 7 years age difference between our kids. The amount of genetic testing from the first kid to the second kid was probably triple, just to look for things that you can find. All right. If we look at double strand breaks, another way you can fix them is with non-homologous end joining. Who knows what's going to happen here? You're just kind of hoping, but it's better than death, right? Okay. So there's no conservation here. You have these various proteins that will bind. I like how we have Artemis in here, named after the Greek god of knowledge, but like I say, I'm not going to ask you guys any detail about this. Just know that this is another methodology for repairing double strand breaks. Double strand breaks are bad. Okay. And here you can see the whole process, but we're just not going to talk about that. Yeah, double strand break in there. Yeah. All right. Yeah, basically kind of fill in or cut off some stuff in there. So ideally, again, keeping in mind that 99% of the DNA doesn't code for anything. If you cut out a few bases here and insert ones that are complementary, so that you can ligate them back together and make your repair your DNA, odds are you're not going to do anything bad, but you might. All right. If we look at site-specific recombination as opposed to our homologous combination, here we have to have specific enzymes. We have these recombinases that recognize particular segments, DNA sequences, and then we exchange them. So our exchange is reciprocal. So we basically cut out like 20 base pairs over here and 20 base pairs over here, and flip-flop them, and you end up making this site-specific recombination. All right. This is kind of the process. We're not going to sweat it. Here we can see a holiday intermediate in there. What happens when this occurs? We can have inversion or deletion. So we can, put a gene in backwards, or we can just, completely cut it out. We can have an insertion. For example, if our DNA is circular, we're just adding something else in there. Okay. Intermolecular recombination is if we're looking at different DNAs here. If our recombination sites are different DNAs. So again, here you can see our inversion. We kind of flip this around and put it in backwards. Our deletion, here we have a deletion, here we have an insertion. Yeah. So we can basically change our DNA sequences through this. What else do we have here? Yeah, how do we create a dynamic genome? If we have our genome, so if we have a circular DNA, we can actually basically double the size in here, but we'll have this Xerxes system that will kind of cleave that in half and get it back to your original size DNAs in there. All right, but that's an odd thing that we have a lot of odd stuff in here. I think one of the last areas I want to talk about are transposons, which they can move genetic elements from one region to another. If you look at our DNA, was it something like 15–20% of DNA is transposons? So they're just basically, many of them carry genes. Some of them have extra genes. You can think about like antibiotic resistance or something there. So for bacteria, we'll have these short sequences that act as binding sites for the transposases that will allow them to insert some DNA in there as well. But transposons are just these nice little sections that can, lift out of one area and insert themselves into, another area in here. And then replication fills in the gaps. Okay, so here we see our target site here. We have these, sticky ends. We stick our transposons in here because they match up with the ends, and then we fill in the gaps here. And now we've, inserted, some DNA into that red section. Okay. So we can think of this, transposition as kind of cut and paste. We can also have replicative, which is our copy and paste. So, we leave a copy behind, but, copy it someplace else as well too. We get our entire transposon being replicated in there. All right, so if we look at direct versus replicative, so here's our direct, we cut it out of here and put it into here. And now we've taken the sequence from the red and added it to the blue. And here we are copying it from here. And it stays in the red, but we also add it to the blue. So we've made a copy and paste, I guess, as opposed to cut and paste. Okay, why is this important? Well, our immune system. If you look at our immunoglobulin genes, our IgGs, we got something like 20,000 genes in here, and yet we make millions of antibodies. So with these genes and the way we can, move all this stuff around, we get our random little sequences in there so that we can recognize all sorts of stuff. So that's how we get the antibodies in our system. All right. So we look at our recombination here, our two heavy and two light chains in there. Those are the kappa gamma. Each has a variable region and a constant region. So the constant region, of course, stays the same. The variable region is the one that's going to be, recombined to give you different sequences to recognize, to produce the different antibodies that recognize the various antigens. Right. So if we look at do a little bit of math here, ultimately, if we have a 300 variable segments in here, we got our four joining segments. That gives you 1,200 arrangements in here. So this isn't precise, so we can get a little bit more variation in here. So we get about 3,000 variants. And then if we have more combinations in here, ultimately you end up with 15 million possible IgG molecules from just a few hundred of these variable segments from our immunoglobulin genes. And this is how we can make our antibodies. And you expose them to different antigens, and you'll make antibodies by just our recombination in here. Okay, so recombination is important for antibody formation. Our actual mechanisms, we're not going to get the mechanism in here. Yeah, I don't know. Do we really want to? No, that's enough of that. All right, that's a lot. So there's flying through chapter 25. And every year I'm like, how can I make these chapters a little bit shorter? Because there's just too many of them. Too much information in these last 5 chapters. Actually, 3 of the last 5 are really bad. But every year I struggle with that. But we'll see what we can do. So let's take a look at chapter 26 and jump into our RNA metabolism. Okay. So RNA metabolism is in many ways similar to DNA metabolism, and in some ways quite different, because we're going to have to talk about capping and splicing as well too. Okay, so our transcription, we're going to see our DNA-dependent synthesis of RNA. We use the DNA as a template to make our RNA. We need to cap and splice our RNA as part of the processing. And then we can look at an RNA-dependent synthesis of either RNA or sometimes DNA if we have reverse transcriptase in there. Okay. Our ribonucleic acids play a lot of roles, but there are 3 main ones that we look at. Our messenger RNAs, our mRNA, they encode the sequences of the to make our various proteins in there. Our transfer RNAs are the ones that match the anticodons to the mRNA while carrying a specific amino acid in there. So we'll talk about tRNA synthase as well. And then our ribosomal RNAs are basically components of the small and large ribosome subunits, and they're responsible for making some of these proteins as well too. But there are dozens, if not hundreds, of types of RNA out there that have a lot of other functions in here. We have a bunch of microRNAs that can help regulate expression. They can bind to things. We'll talk about like silencing RNAs, siRNAs and SORNAs and oRNAs and nRNAs, and there's all sorts of crazy stuff out there. And of course, we have to keep in mind that some RNAs have catalytic activity and can function as enzymes. So we classify them as ribozymes because they're ribonucleic enzymes. Okay. And of course, if we look at some viruses are their genomic material is made up of RNA instead of DNA, okay? Which of course means it's probably highly mutable, so harder to treat them. Okay, so where does RNA come from? It gets transcribed from the dNRNA. So transcription is tightly regulated, so we can control the concentration of each protein. Okay? We've seen all this with our regulation before. Man, is it cold in here. I need to build a fire. I miss when we actually had like a little Bunsen burners. But anyway, ribozymes are mostly single-stranded. We're going to talk about how RNA folds as well too. They can fold into particular structures, and that folding will enable specific functions. Right? Some RNAs act as catalysts, and then we'll see our processing in here. We're going to talk a lot about mRNA, talk about splicing, polyadenylation of the 3' end, and capping the 5' end. All right. And again, we start in bacteria, and then we move our analogy into eukaryotes. So if we look at E. coli, the nucleotide triphosphates add to the 3' end of the growing RNA strand. So just like we saw 5' to 3' for DNA replication, we see 5' to 3' for our mRNA transcription as well too. Growing chain is complementary to the template strand in DNA. So the synthesis gets catalyzed by the enzyme RNA polymerase and involves, magnesium ions again, because you have those large phosphate groups in there. So the magnesium helps diffuse that big negative charge. We're actually going to see our RNA polymerase is going to bind to our DNA, unwind about 17 base pairs worth of it, and it's going to cover about twice as much as that. Okay. So here's what we see. Here's our template strand up here in pink. Our growing RNA strand, you can see that instead of having thymine, we have uracil here. And so we grow that just like with our DNA replication. We have our DNA polymerase. Here we have an RNA polymerase that rolls along here and makes RNA from this. Okay. Yeah, check something real fast here. Make sure that it's not. Okay, good. Okay, awesome. All right. So the features of transcription, we have to have a promoter sequence to bind to. Okay, so we don't need a primer, but we do need a promoter where the RNA polymerase can bind to. And in the growing end, we'll insert base pairs in there, inserts 50 to 90 nucleotides per second. And we make this nice little 17 base pair bubble as it slides along. So if we have our, I'm prepared, I got the fancy stuff here. If we have our, awesome double-stranded DNA, we actually make like a 17 base pair. Let's see if I can hold these. Little bubble in here. Something I can't. So we can make a bubble that slides along there, making our mRNA, and then the DNA can close up behind that. The neat thing is you can actually have multiple RNA polymerases on a single strand of DNA so that you can be making lots of mRNA from the same sequence simultaneously. Okay. All right, so here's our transcription bubble pictorially. Unwind a little section here. Here's our RNA coming out, and then wrapped in here would be all this will be inserted into our RNA polymerase. Okay, like so. All right, nucleotides come in. Growing mRNA chain comes out this way, and here we see our unwinding bubble of our DNA. So we end up with positive supercoils on one side, negative supercoils on the other. Okay, so if we look at this, here's our DNA double strand. You can see our complementary sequences here. We look at our 5' to 3' RNA transcript. It is going to look exactly like the 5' to 3' DNA template or coding strand, except instead of thymines, we're going to have uracils there. Okay, so this is our coding strand, and this is our template strand or non-coding strand. Okay. Why are there 3 stars there? That's funky. Okay. So both DNA strands can code for proteins in bacteria in here. So we can either have top strand or bottom strand. Not that it really matters. Adenovirus has a linear genome, and each strand codes for proteins. So here you can see our adenovirus, and depending on which way you go, you get different proteins in there. It is kind of neat that there's only a little bit of overlap. All right, some palindromes, I guess. RNA polymerase is a very large enzyme, has no proofreading capability. Okay, so it makes a lot of error in there. All right, but that's okay because if we make a bad protein, we can just ubiquitinate it and chop it up into little pieces. We're not going to talk about all the different subunits in here, but they all play particular roles in there. Various promoters, and again, keep in mind this is all an E. coli for now. You're going to have your TATA box, your TATA sequences in there, and they are consensus sequences where the promoters will bind and allow the attract the RNA polymerase to it. Also, somewhere 40 to 60 base pairs upstream of the initiation sequence, you have these other promoter elements. Okay. The AT sequences promote strand separation. Why? Because they only have 2 hydrogen bonds pairs in there, as opposed to GC, which has 3. So it takes less energy to break AT-rich sequences. So that promotes the strand separation. So you can start making that transcription bubble. Okay. All right. Oh yeah, and just anything here, where we start, the first nucleotide of the RNA molecule is our start site, but we have things before that like our promoter site and stuff like that. That's why we have negative numbers, because they are upstream of the actual mRNA site. Okay, so upstream elements, our Tata boxes, we have these little spacers in here, but here's where we actually start making our mRNA. Okay. All right, and these are just various things that can bind. All right. So we can use this neat little footprint technique to find DNA binding sites. So you can isolate a DNA fragment, radio label the DNA, bind protein to it, and then keep another as naked DNA control. So you've got one with protein bound and one without it. And then you treat both samples with something that cleaves the DNA, you separate the fragments, and then visualize the bands on a plate. So what you end up with is, hey, our missing bands here, well, this must have been bound to something. So we can say, here we've cleaved our protein, but here we're missing these sites. So all this DNA in here would be where our polymerase was bound. Okay, so we can say, hey, there must be a binding site in this region of DNA. Okay. All right, that's just how we figure out. And again, we can see little sections here where there must be a polymerase bound. All right. So take a look at my time real quick here. Good. Okay. So let's talk about the important stuff here. Okay. So, first we got to figure out where it's going to bind and what's going to happen, but we're going to do initiation, elongation, termination. Okay. So our polymerase will bind to its promoter, and this sigma factor will bind. So our sigma subunit binds in there too, and it creates a closed complex where we haven't yet unwound the DNA. We're going to start unwinding it a little bit to make that transcription bubble in there. So that's when we make our open complex. And then our polymerase is going to start chugging along down the promoter. Okay. So elongation, the polymerase binds to the triphosphate nucleosides and generates the mRNA transcript. It starts adding those on that 3'. We kind of slide our along the DNA, and our growing RNA strand gets ejected out there. Right? I'm probably gonna get splinters doing that from these old things here. Picture's worth 1,000 words. So here we are binding. We open this up, make our transcription bubble, allow our nucleotides to come in. We initiate transcription. We start, and you can see here, we have a complementary sequence. Our elongation, we start sliding along here and moving the mature mRNA out. Okay. Eventually, we get to our termination site, and then we release our RNA, and we fall off of the DNA strand as well too, and then go back on and do this again. Okay. There are 7 sigma subunits in E. coli. I don't know, you don't really need to know that. There's a lot of them. How do we regulate this? This takes a lot of energy because we're spending ATP to do this. So it's energy intensive. So we're going to regulate it, and we can regulate it in lots of ways. One way is we can modulate the affinity of the polymerase for the promoter. So we can have promoter sequences that differ from the consensus sequence. Right, so it's like, yeah, it's close enough. It's kind of like you found a parking space. It's like, I think I can fit in that one. Or if you ever tried to park in like a truck parking space, like, yeah, I can park in here sideways. So how well will our polymerase fit in there? How well will it fit in there? Okay. We can have various activator proteins that will enhance the binding activity of the polymerase for a particular sequence. And we can have repressor proteins that will block binding sites, right, and prevent them from binding at all. Okay. There are 2 types of termination sequences for E. coli. We have a row-independent one. Okay, so what we tend to see is a series of 3 uracils near the end. And what happens is there's a self-complementary region that forms in here. So you make this little hairpin, and this little hairpin acts like a speed bump. The RNA polymerase pauses, and it's like, yeah, I didn't really want to do anymore the same way, and kind of falls off, and that's where we're done. So that's our row independent. It's basically just depends on the sequence of the mRNA of the RNA that's being made. We can also have a row dependent, where you have this row helicase that comes on here and binds, and this binding, as our row helicase migrates along the mRNA, in there, it will actually separate the mRNA from the DNA template and cause it to fall off. So there's row dependent and row independent, two different mechanisms. Okay, let's compare this with eukaryotes. It's the same, but more complex. We've got, again, multiple RNA polymerases, 1, 2, 3 in here. So polymerase 2 is probably the one that we're going to talk about most because it synthesizes mRNA. 1 makes these pre-ribosomal components, and 3 makes tRNAs and some other small RNA products in here. There may be a polymerase 4 that makes these siRNAs that are particularly used in plants. We can use them in people, but plants, I believe, actually use them on their own. And of course, mitochondria have their own RNA polymerase as well, too. So we can call that mtRNA polymerase for mitochondrial, mtRNAs, mitochondrial RNAs. We do see something similar. There is a TATA box. Note that it's about 30 base pairs upstream as opposed to 10. We have an initiator sequence in here, and then we can have various regulatory sequences, a few dozen to a few thousand base pairs away. They can be really far away. Anyway, we're going to have a lot of proteins in here. So if you thought there were a lot of proteins and subunits involved for bacterial RNA transcription, it gets worse here. So there's a lot of protein-protein interaction to drive this transcription. Okay, here you can see a whole bunch of them. We have our transcription factors, TF2A, TF2B, so on and so forth. All sorts of good stuff in there. Anyway, how do we start? We initiate by having a TATA binding protein, abbreviated as TBP. It will bind the promoter in there, bind with the promoter. And so as it binds the TATA box, you see this promotion, which will attract in our other components in here. Our TATA binding protein is actually part of a multi-subunit complex. So you've got, all of our various transcription factors in here. These can function to help unwind the DNA. We can phosphorylate the polymerase at its carboxy terminal domain, which will change its conformation, and that lets RNA polymerase 2 get to work. Okay. As we start, eventually transcription factor 2E gets released, and then so does H, and our elongation factors bind, and they enhance the processivity as it chugs along the DNA strand. Okay. Some of them are bound to the phosphorylated carboxy terminus domain. For termination, we dephosphorylate, and that allows it to fall off. I like this understatement. Regulation is complex. There are just layers upon layers upon layers of how all this stuff interacts with each other. And it's like, how fast do we dephosphorylate? How various it's like the bacterial, but just layers of complexity on top of it. Okay. But the neat thing is, here's our start site, our initiation. We have this pre-initiation complex where we get everything to bind in here, and then we open up the DNA, and we begin our initiation. We go chugging along. We have our various elongation factors that help promote this to keep going along here, and eventually we'll have termination factors that will come in. We'll see that dephosphorylation, and various components will fall off, and then we'll be done. Right? Okay. Transcribed genes are more actively repaired than silent genes. So, we may be, replicating these genes, but the ones that are transcribed will be repaired more often because they tend to have more active products. And, we can see that various genetic diseases can be associated with defects in TF2H. Okay. We can work to selectively inhibit some RNA polymerases. So if you look at various antibiotics, things like actinomycin or rifampicin, they will bind and inhibit bacterial synthesis in here. If you look at the alpha-mannitine, which was referenced way early in the book someplace, this actually blocks polymerase 2 and 3. The neat thing is it doesn't block its own polymerase 2. So, it will kill anything that tries to eat the mushroom, while allowing the mushroom to continue on its replication. So that's pretty neat. Pretty horrible. Yeah, don't eat strange mushrooms that you find in the forest. Okay. Let me take a look at something here. Yeah, we got time. We got lots of time here. Where are we at? Oh, good. Okay. Actually, this chapter is not as long as I thought it was. So, okay. So what we have here now is we can talk about the processing of mRNA. Okay. So we've transcribed RNA, but we're not done. It's kind of like, we've built our car, but now we got to paint it and put in the upholstery or whatever. Something. Terrible analogy. So processing includes things like splicing out the introns and rejoining our exons to make a continuous sequence, adding a 5' cap and a 3' poly A tail. And of course, we can degrade the RNA as well, too. Okay. So our 5' cap is just telling us, hey, typically we're going to see a methylation involved in this. Okay. And it just tells us here's the start of our RNA. It kind of maybe sometimes adds to its stability a little bit. But we complete this. We also, once our polymerase is done, we'll add a whole bunch of adenosines at the end as well, too, in there. And now we can splice out our various introns and leave our exons here with our 5' cap and our poly A tail. And we now have a mature mRNA that is ready for translation. Okay, so what is the 5' cap? Typically, it is a methylguanosine. So I always think of like those little berets, or put a little methyl on it, make it jaunty. So we methylate that initial guanosine in there. So we have our methylguanosine. We can see additional methylations at other sections in there, from the following ones in here too. But we're typically going to start with this methylguanosine in here. What this does is it helps protect the RNA from various nucleases. So it enhances the stability so that RNA can survive for a bit. It also forms a nice binding site for the ribosome. So that methylguanosine is important for interaction with the ribosome. Our introns are found in most genes. Exons are typically about 1,000 base pairs. Our introns can be anywhere from 50 to 700,000. So you can make this huge RNA structure and cut out 99% of it to make your mature RNA. Some genes have dozens of introns. And again, we kind of saw with like POMC, you could get like, was something 11 different protein products based on the splicing that's involved in there. Okay, so our introns are going to get spliced out. We've got 4 classes of introns. We have group 1 and group 2 introns. They are both self-splicing. They will automatically cut themselves out and leave. They differ in the splicing mechanism. We'll look at that here in a minute. We have spliceosomal introns, which we need a spliceosome complex in here that will come in and cut out a particular sequence. So they're way more complex than our group 1 and group 2. And our tRNA introns have these protein-based enzymes that will splice them out. All right, and there are particular endonucleases that will recognize particular sequences to cut those out. All right, so they're a little bit simpler. So group 1 introns, basically, you have a free guanosine on one side here. This free guanosine, the 3' hydroxyl on there is going to act as a nucleotide and attack the phosphate at the 5' hydroxyl splice site there. Okay, so there's our splice site. We can attack this way. Yeah, it's going to cut out this way. So this cleave this out, and then our hydroxyl group here will attack this one, and we just end up cutting out our spliceosome here. The important thing about this is we have a linear intron that gets spliced out. Here's the mechanism in here that looks at this way. So we have this left here, and then it's going to go over and attack this way. Okay. Compare this with our group 2, and we see a different splicing mechanism that occurs here. Here we're going to have a sequence inside of our intron that will actually function as the active hydroxyl group and attack the splicing site. And what you end up doing is you form this lariat structure. So a lariat is like our little lasso from the cowboys, like yeehaw, get on little doggies and whatnot in there. So we form this lariat structure, And then our hydroxyl group on our primary transcript can attack here at the other end of the splicing site of the intron, and then it will cleave that out. So you'll have this little, lariat that ends up leaving, and you make your spliced RNA that way. Okay, so group 1, you get a linear section. Group 2, you get a lariat section that comes out. Okay, spliceosomes have this huge super complex made up of SNRPs, or Smurfs because they're blue. Okay, no, SNRPs, small nuclear ribonuclear proteins. I mean, how many times can we put nuclear in here? We can have fun with this. It's small nuclear ribonuclear proteins. Anyway, our SNRPs, our SNRP RNA is called snRNA for small nuclear RNA. They're short. There are 5 known ones: U1, U2, U4, U5, and U6. Where's U3? I don't know. It's just a crazy naming there too. But typically, we'll see these particular sequences here that will mark splicing sites, and then these SNRPs can interact with them and induce the cleavage in there. Okay, and we'll see some really complex interactions here. So keep in mind, this is all single-strand linear nucleic acid. So we're going to have a lot of funky folding and interaction between our various SNRPs that can bind to our RNA and induce that cleavage in there. Okay. We're not going to go into the massive stuff that goes in here, but again, our SNRPs end up in the formation of a lariat, and they leave with the lariat. So as opposed to our self-forming lariat in group 2 introns that just kind of gets tossed out, here we have a bunch of cowboys, I don't know, U1 through U6, that come in and make the lariat and then take it away. Okay. And then our tRNAs do their thing. All right. Let's talk about the poly A tail briefly as well too. So when we add this poly A tail, we are going to actually have our RNA polymerase 2 synthesizes it beyond this site here. So the cleavage signal gets bound by an endonuclease and a polyadenylate polymerase that is bound to that carboxy terminal factor in here. So here we see this polyadenylate polymerase, which is just going to say start adding adenines, and so you get A. I don't know if you've ever used like the if you've ever done any genetics work where you got to look at all these sequences. It's a pain in the butt. So having your computer actually speak it and read it is pretty handy. I can remember when I was making site-directed mutations in some bacteria, and I was checking my sequences. So I'd have like my little piece of paper here to check and make sure everything was right. I just had the computer read it off to me. So you get to poly A tail, it's like A, just that monotone voice reading off letters to you. Anyway, no point to that. I'm just rambling. But typically, we're going to see our poly A tail be somewhere between 80 and 250 nucleobases in there. All right, so if we look at our massive overview here we have our DNA that gets transcribed into our RNA. We have it put on our methylguanosine cap, and possibly a couple other methyls in there, but the methylguanosine is going to be the start form. And then we have our poly A tail here as well. Okay, we splice out our introns through whatever mechanisms that we have. Okay, and that means we'll also end up cleaving off a bit of the poly A tail as well too, but we retain some of it. And then we end up with our mature mRNA with our various exons in here. Now we can have alternative splicing. We don't have to have L1, 2, 3, 4, 5, 6, and 7. We might leave out 3 and 6 and get a different mature mRNA that encodes for a different protein. Okay, so that leads us into our alternative splicing. All right, so maybe we don't put all those exons in there, and something like 95% of human genes are alternatively spliced, so that we can make either isoforms or completely different proteins as well too. Okay. One of the neat things is our poly A tail form can sometimes have different sites where it forms here. And this gives us, again, greater diversity. So we can look at this again with our immunoglobulin chains, our IgGs, with all that variability in there. If we have these multiple poly A sites, that can enhance genetic diversity. Okay. All right. If we look at, say, the calcitonin gene of rats, okay, so here we can see in the thyroid, we splice one particular way, okay, and, to get, well, we have a polyadenylation at one particular site, so we end up with a shorter total mRNA. Then we splice this out, and we get our mature mRNA, which is different. We make our calcitonin. If we have a calcitonin gene-related peptide in the brain, okay, here we go a little bit further. So we have a longer mRNA, and then our splicing includes, it doesn't include 4, we cut out 4, but we have this 5 and 6 that are added in here, which gets translated, and we end up making our CGRP protein. Okay, so the first half of both of these is the same, but the second half, different sequences with slightly different functions. All right. Let me take a peek. Where are we here? Time-wise, oh, okay. Let me take a drink and we'll keep going. Feel free to interrupt with questions at any time. I'm just cruising on here because, we go ahead.
Oh no, I was gonna say okay.
Yep. I just need to pause for a drink. I'll give you guys since I knew it was going to rain today, I was like, crap, I better mow my yard yesterday. Stirred up all the pollen. I actually had a really good mask. I had like a big handkerchief that I soaked in water. It was awesome, except like the last 15 minutes when it dried out.
Mm-hmm.
That's when I inhaled pollen, so my voice is a little, yeah.
Yeah,
You guys know. It's North
I
Carolina.
definitely understand.
Okay. So let's see if we can get through this. Let's talk about, mRNA is probably our main focus in there because that's, our active protein form. That's probably our, what we talk about the most. tRNA is maybe our second most important. And we'll talk about ribosomal RNA a little bit as well too. So when we process our tRNA and our rRNA, we actually get a bunch of modifications in here. So we're going to incorporate, pseudouridine, thiouridine, dihydrouridine, and all sorts of, unusual bases in here. So here's a thiouridine where we have a sulfur molecule in here. We can see inosine will get some incorporated, methylguanosine. You can see all these various different compounds in here. Pseudouridine plays a pretty important role. So, talk about it a little bit in here, but we have a bunch of, modified bases. So it's not just our GCAU in these RNAs. We have these other things that are in there. Our rRNAs and tRNAs, much like our mRNAs, get cleaved from longer precursors in here. So here we see our pre-ribosome. We cut this down to maybe our pre-40S and our pre-60S forms, and then we cut those down to 40S and 60S, which are made up of these different components in here. All right, the important thing is, here we see our primary transcript. Here we see our intermediate, okay, where we've now started modifying these. And then we cut out the sections in yellow, okay, so that we get our mature tRNA, okay? And this tRNA will code for tyrosine. You can see our ACC up here on one side and our codon section, I believe, down here. I'm not sure exactly where. We'll have to take a look at that in a minute here. Various microRNAs function as well too. So these tend to be very short pieces, segments of RNA, typically maybe like 22 nucleotides, and they will bind to mRNAs to alter the translation. So these can help cleave mRNAs, or they can block them from translation as well too. There are something like 1,500 genes encoding these microRNAs, and one or more of them affect the expression of most protein coding genes. So, these play a big role, and they're not super well understood, but they are synthesized from larger precursors, which are processed by two specific endoribonucleases, DROSA and DICER. DICER you may have heard of because we use it to cut these RNAs in there, but it's found other uses, particularly looking at maybe like gene therapy in there. And then it's kind of neat because DICER can make us double-stranded RNA, which makes it maybe a little bit more stable in there as well too. And Anyway, we can take all these microRNAs and load them onto protein complexes. Like this RNA-induced silencing complex, or RISC. There's a RISC we'll never hear from that RNA, but basically this RISC complex will bind to the target mRNA and prevent it from undergoing translation. Okay. All right. And then if it's a perfect complement, then we cleave the mRNA and it doesn't get translated. If it's only a partial complement, we got some ends hanging out or some little gaps in there, then the translation is just blocked. All right. So it seems like a lot of effort to make this huge mRNA and then cleave out a bunch of it and then have a small segment come in there, bind to it perfectly, and then just chop it all up. But that's a regulatory process. Okay. So, and this shows us part of our methodology of processing to make our microRNAs. Okay, that can then come and bind to our mRNA and lead to its cleavage, or partial complementarity, where we have the little gaps in there where we just kind of slow it down, block it. Okay. All right. Ribozymes have enzymatic activity themselves, so they can either have autoenzymatic activity where they cleave themselves, or they can function on another RNA molecule as well too. Again, these follow Michaelis-Menten kinetics. Didn't think you could escape chapter 6 forever, right? So we could talk about KM and competitive inhibition and all that good stuff in there as well too. But probably the main thing to understand is that we have nucleophilic attack from our hydroxyl group on the phosphate, which will allow us to hydrolyze that phosphodiester bond and induce a chemical reaction. Okay. Some of the well-characterized ones are self-splicing group 1 introns. Those are a great example of that. We have something called the hammerhead ribozyme because it looks like a hammerhead shark. This 2' structure, looks like a nice little hammer in there. And then RNase P cleaves precursors to tRNAs by recognizing the shape of the pre-tRNA and the particular CCA sequence in there as well too. But those are ones that we know fairly well. So here's our hammerhead. Kind of looks like a hammer up there a little bit, I guess. Strange. Of course it folds, so it doesn't really exactly look like that. I don't want to try and look at that. Once we have our mRNAs, presumably they actually make it through translation, or even if they don't, we can control how long an mRNA exists. So if we degrade it fast, that's one way of regulation. If we let it stick around for a long time, it's going to make more protein. So that's that lifetime of our mRNA is our a method of regulation here. So each gene product is needed for a certain amount of time, depending on what it's making. Those half-lives can be anywhere from seconds to hours. So we can control the rate of synthesis versus the rate of degradation. We can look at our nice little steady states, because again, in an ideal world, we're going to have this homeostasis going on. But typically our vertebrate mRNA is going to last around 3 hours. We'll see about 10 turnovers per cell generation. Bacterial mRNAs have much shorter half-lives, like on the, like 100 times shorter. 2 orders of magnitude. There we go. How's that? Our various ribonucleases will degrade these. So you always hear the thing about DNA. DNA is forever. You can convict criminals from the 50s and 60s based on DNA samples. But RNA, you look at it funny, it disappears. That's because our ribonucleases are everywhere. Your fingertips contain them. You constantly, there's just tons of them here. So how can we make RNA stable? Well, there are ways that you can try and promote it in there, but you can make these various hairpin structures that can add stability. The capping and the tail aid in the stability as well too, so that they'll stick around for a little bit longer. But typically, in nature, they're going to encounter some RNases somewhere pretty quickly and get degraded. You can keep them in water for like a pure water for a very, very, very long time, but yeah. Anyway. We can also do something else that's kind of funky, and this was a major discovery that got a lot of press when HIV was first discovered. So there's reverse transcriptase, where we can actually kind of cheat with our central dogma, and we can have RNA that replicates itself, and then it can reverse transcribe itself into DNA to incorporate into, say, a host chromosome. Okay, so the virus enters the host cell, reverse transcriptase makes DNA from an RNA template. It'll degrade the RNA, because that's very easy to do, and then you replace it with double-stranded DNA that can get incorporated into the host DNA. Okay, so if we look at a typical retrovirus here, it binds to the plasma membrane, releases its RNA genome in here. We see our reverse transcription. We make our DNA. We integrate this into the host chromosome. When the host chromosome is replicated, you actually, get a transcription. So that transcription will have a signal sequence on it. So it'll take it out to the ribosome in here, where we'll make copies. We'll translate it, and then we'll also see proteolysis. So we can, we basically our RNA genome will contain copies for binding, infection, and our various viral protein particles in here, as well as our reverse transcriptase. So we'll see this assembly of our viral capsid and then lysis of the cell. Our new viruses can go on to affect neighboring cells there. All right. So typically we have three important genes in retroviruses. Our GAG, which is our group-associated antigen. So this is going to be our viral core. We have a PAL. This is going to increase our protease, encode our protease that cleaves the reverse transcriptase and it integrates to insert it into the host enzyme. And our ENV, our envelope proteins in here. And then we'll have typically long terminal repeats that make it easier to get it into the host DNA as well. Okay. So here's what we see. So we're talking, a few hundred base pairs could be the bare minimum necessary to make a functioning virus, which is a quasi-life form. All right. We can actually generate viruses. This would be something we should do this in a lab. We should make a virus. That'd be wrong. I don't know if you guys ever played Plague or one of those online games where you try and craft a disease that kills the entire world. That's when you got to close down the ports in Madagascar or whatever. But I should link that. I think I used to have that on here, but I need to find that game. I'll put that on in the module there. But it kind of gives you a sense of how various infectious diseases can occur and adapt and infect populations. Anyway, but this is our bare minimum that we need for a virus, a reverse transcriptase virus. Typically, our reverse transcriptases, we got three reactions: RNA-dependent DNA synthesis, RNA degradation, and DNA-dependent DNA synthesis. Okay, so here we make a complementary DNA sequence to the RNA. We degrade the DNA, and then we make a complementary DNA sequence to the DNA that we have left behind. Okay, so we'll have a zinc in here. We'll have a tRNA primer. And typically we see no proofreading mechanism in here. This is one of the things that made treating HIV so pernicious, because it had no proofreading, so it made tons of errors. So it was very hard to develop a drug that would bind to HIV, because it was constantly changing its sequence. So that's where the three-headed method of fighting it kind of came in there. All right. Some retroviruses will also encode oncogenes as well too, and these can basically lead to cancers. So was it the Ruth sarcoma virus here? Is one of these examples. So here we just see a little something extra added in. Yeah, it's like, hey, besides a viral infection, you get cancer, yay! HIV causes AIDS. So we have our reverse transcriptase is particularly error prone. So we have all this sort of stuff in here. It makes it really, really hard to vaccinate it. Although it's fascinating. I actually have a friend who is in the PrEP-X trial and has been for years. So there is potentially a vaccine for HIV out now and in testing. So, but anyway, that's what 40 years after the onset of the AIDS pandemic. It's I remember when there was actually a show recently too about the onset. It used to be a death sentence. If you got AIDS in the 80s, it's like, you're dead. So there were these life insurance policies and some scams of bundling those and selling them. And the idea was, you sell all these life insurance policies for people who had developed AIDS, and then you'd collect on the life insurance policies really fast and make a whole lot of money. Well, as medicine got better and better, and lifespan, it's now, you think about AIDS, and you probably know people who have AIDS who are living with it fairly normal lives, and it's now for most people an inconvenience, but it's something that they can live with. So there was a huge Ponzi scheme of these, like some documentary about the guy who came up with this idea and how he had, it's like multimillionaire, and then just went completely broke by trying to scam people out of money for these insurance policies. I'll have to look that up. Maybe I can post a link to that too. Anyway, we do have some various targets that we can look at here. We can target the reverse transcriptase itself. We can try and inhibit that. We can have our various protease inhibitors as well too. So you'll see like your avirs versus your genes or zines in there. And the combination cocktail treats that pretty well. One interesting analogy, our retrotransposons in eukaryotes are very similar to retroviruses because they are mobile genetic elements in our eukaryotes that can move nucleic acid sequences. They basically move positions in the DNA using an RNA intermediate. Okay. So these are unlike the bacterial transposons that move DNA directly to DNA. Here we use an RNA intermediate to do that. Okay, so you can see our retrotransposons, look something like this. So that they don't have an envelope gene, so they don't make viral particles, but they still have the gag in the paw and the long terminal repeats in there to help move stuff around. Okay? And you got a place where you can stick stuff in there, other DNA in there. Okay, our telomeres are the structures at the ends of our chromosomes. They have tandem repeats. We sort of talked about these a little bit. They can be tens of thousands of base pairs long in mammals, but you lose a little bit of ends each time you go through replication. Okay, so we have this telomerase that will extend the ends of chromosomes in here. So we can, our chromosomes get shortened, the telomerase adds telomeric sequences to kind of solve this problem. There's a theory that if you, upregulate telomerase and keep adding that, you can, essentially get cell immortality because, what was it, that Hayflick limit, you can only divide about 50, 75 times. I forget exactly what it was there before you start cutting into the actual important stuff and you can't replicate anymore. With telomerase, potentially you can avoid that and actually have infinite lives. It's kind of like Doctor Who, supposed to have only like 12 regenerations, and now there's hand waving because we got to have another actor in there. Anyway, there were some studies with mice that their lifespan, their telomeres seem to have a stable length with a particular telomerase type therapy. All right. Finally, we can talk about an RNA world. If we go back to evolution, you would say what compound came first? DNA, RNA, proteins. There's some support for RNA being the first because it is very abundant. RNA has catalytic activity and it can hold genetic materials well too. It's not super stable, but it exists in the forms that both DNA and RNA are particular suited for. Like you can't store information in protein, you can't have catalase activity in DNA, but RNA is kind of the bridge between those. So here is just kind of the support for saying you could start with RNA and develop life from that. You can look at the various types of reactions they can catalyze. These peptide bond formations, a lot of non-living entities. It's interesting because is a virus alive or not? And here we're classifying it as non-living. I tend to say quasi-living because it can't really live on its own, but it can reproduce. So as long as it has a host cell in there. So we'd have to ask, is there a ribozyme that directs its own synthesis in there? Okay. Whew. So there you go. There is a super fast jaunt through chapter 26 and our RNA metabolism. Um, I tend to find RNA metabolism a little more palatable because it's much more directly focused. Our DNA metabolism is like, well, you got all this other stuff that can happen in there. So basically, I want you to know the basics of that and maybe a little more details of this shorter RNA chapter as well. Okay. We will pick up on Wednesday with the next chapter, chapter 27, where we'll talk about protein metabolism. Yay! And then we have one last chapter after that, and then we're done. Um, I know I'm going super fast here, product of our short semester, but take a look at your um, um, homework online. Make sure you check out the homework online. I think there's like 8 to 10 questions from each of these chapters, which they show the basics of what I want you to know. And like I say, the final exam will be very similar to exam number 3, which I'm still working on. I should have those grades posted, if not tonight, tomorrow. So, all right. Any questions about any of that? Did I bore you to death? Did I scare you to death? All right. Well, even finished with 1 whole minute to spare. So I'll let you go and I'll post this recording as soon as it becomes available. So I will see you on Wednesday.